This recording is for the second half of cellular respiration, the Krebs cycle, the electron transport chain, and fermentation. So as you'll remember, at the end of glycolysis, we made two pyruvate molecules. So after glycolysis is finished, and if oxygen is present, the next step in cellular respiration is the Krebs cycle. The Krebs cycle occurs in the mitochondria. And you should know that the mitochondria has two membranes, which are called the inner membrane and the outer membrane. So where does pyruvate go? After glycolysis, pyruvate is transported from the cytoplasm. Remember, that's where glycolysis took place, within the cytoplasm. So pyruvate goes from the cytoplasm into the mitochondrial matrix. In the matrix is where enzymes are present. Remember, enzymes are catalysts. You know a word is an enzyme if it ends in ASC, and a catalyst speeds up chemical reactions. Acetyl-CoA is going to be initially produced from pyruvate in the intermediate step, and then acetyl-CoA will go into the Krebs cycle. So I'm going to read and discuss the Krebs cycle from page 133 in your textbook. So I told you where you see glycolysis, which occurred in the cytoplasm, with its products with two pyruvate molecules. After glycolysis, you have the intermediate step. The formation of acetyl-CoA is that intermediate step. The pyruvate has three carbons. Pyruvate reacts with NAD and coenzyme A, which you see as CoA, to produce NADH and carbon dioxide. The big major product from this is acetyl-CoA. So in your book where you see that carbon dioxide coming off, every time you have carbon dioxide coming off, you lose a carbon. So we have a 3-carbon molecule pyruvate, which is reduced to a 2-carbon molecule with coenzyme A attached. So 2 carbons plus CoA is acetyl-CoA. Acetyl-CoA is the product of the intermediate step, along with NADH and CO2. So in your book, you'll see where it says first stage, pyruvate is split to form carbon dioxide and an acetyl group. The acetyl group is just 2 carbons. The acetyl group attaches to CoA, which is coenzyme A, to form acetyl-CoA. At the same time, simultaneously, NAD receives 2 electrons and a hydrogen from pyruvate to make NADH, and then acetyl-CoA goes into the Krebs cycle. Remember, NADH is an electron carrier, and electron carriers carry their electrons to the electron transport chain. So acetyl-CoA is going to react with oxaloacetate, which remember begins and ends the Krebs cycle. Acetyl-CoA reacts with oxaloacetate to make citrate. Now, in this reaction, coenzyme A is released, and water is going to react to donate a hydrogen to the coenzyme A and oxygen to citrate. So, acetyl-CoA plus oxaloacetate plus water yields CoA plus citrate. Now, citrate is a 6-carbon compound. Citrate is automatically converted to isocitrate. Isocitrate then reacts with NAD to make NADH. A carbon is released in the form of carbon dioxide, and then you have alpha-ketoglutarate. Now you'll notice we started with 6 carbons in isocitrate, but we lost some of those carbons, so now we're down to a 5-carbon molecule. Now alpha-ketoglutarate reacts with NAD and ADP to form NADH. Another carbon is lost. We have carbon dioxide, ATP, and succinate. So if you're going to write that out, alpha-ketoglutarate plus NAD plus ADP yields NADH plus CO2 plus ATP plus succinate. Succinate, we're down to 4 carbons now because we lost 1 to carbon dioxide. 4 carbons, succinate reacts with FAD to produce FADH2 and fumarate. Fumarate reacts with water to form malate. So you see over in step 6, fumarate is converted to malate, which contains 2 additional hydrogens and 1 additional oxygen derived from water. So the whole purpose of water coming into this equation was to give us its hydrogen and its oxygen. And when that happens, we form malate. Malate reacts with NAD to make NADH and oxaloacetate. Oxaloacetate begins and ends the cycle. So you're going to have to label what you see on page 133, and you're going to have to write out in words what is going on. And those words are written for you up under the first stage and second stage. We also write them in class, but basically if you write through what I've just talked through, then you'll know what you need to know for your test. For every glycolysis step, you get 2 pyruvates. So the Krebs cycle turns twice for every glycolysis process. So what you see here is times 2. So in the Krebs cycle, really you have 2 NADHs formed in the intermediate step because 2 pyruvates entered in from glycolysis. Then in the cycle itself, you have 2, 4, 6 NADHs formed. You see 3, but say that times 2. You have 1 ATP you see there, so that times 2, so 2. And FADH2, you see 1 there times 2, so it's 2. So the things to remember, every time glycolysis happens, you have 2 pyruvates formed, and those 2 pyruvates are going to enter into the Krebs cycle one after the other, so the Krebs cycle keeps turning. Remember, that's why it's called a cycle, because it keeps turning. It begins and ends with the same thing, oxaloacetate. So just reading down there at the bottom of page 133, for each acetyl-CoA, the Krebs cycle produces 2 carbon dioxide, 1 ATP, 3 NADHs, and 1 FADH2. Now that's just for 1 acetyl-CoA. The formation of each acetyl-CoA prior to the Krebs cycle also generates 1 carbon dioxide and 1 NADH, and there they're referring to the intermediate step. So for each pyruvate molecule supplied by glycolysis, the mitochondrial matrix reactions produce 3 CO2s, 1 ATP, 4 NADHs, and 1 FADH, because each glucose molecule produces 2 pyruvates. That's the key. Remember, 2 pyruvates enter in from glycolysis. Now, we did not make a lot of ATP in glycolysis, and we did not make a lot of ATP in the Krebs cycle. What we did make a lot of was electron carriers. NADH and FADH2 are both electron carriers. There are a ton of different animations on YouTube, or you can Google them. Find one that helps you the most to go through the Krebs cycle. If you're still not getting it from the book and from this, there are so many animations that you guys can look at for your test to help you study the process of the Krebs cycle. Now the electron transport chain, that's on page 134, and in the electron transport chain, the NADH and the FADH2s that we made in glycolysis and that we made in the Krebs cycle are going to enter into the electron transport chain. You're not going to have to label the electron transport chain on your test. What you're going to have to do is be able to answer multiple choice questions about it and be able to write out the steps. So the steps of this are actually pretty easy. The big things to remember. The whole purpose of NADH and FADH2 is to transfer their electrons, and along with that, to transfer their hydrogen. So the first stuff in your book, NADH and FADH2 donate their electrons and hydrogens in the electron transport chain. So see, first on that picture, NADH comes in first. NADH drops off its hydrogen as well as its electrons to become NAD. The energy of those electrons flows through the rest of the chain. When it drops off its hydrogen into the intermembrane space, what we're going to get is called a concentration gradient. FADH2 does the same thing. It comes in and drops off its electrons and its hydrogens, and a concentration gradient occurs when you have a buildup of hydrogen, buildup of something, hydrogen, sodium, calcium. whatever it might be, on one side of the membrane. To get that buildup, and on the other side, they're not there. So that buildup builds up pressure, and that pressure has to be released somewhere. What they say is that the hydrogen concentration gradient creates a potential energy situation. Potential energy has the potential to do work. In this case, it's converted from potential energy to chemical energy in the form of ATP. So that buildup of those hydrogen ions is crucial to making ATP. So we have that buildup of hydrogen ions, and what's going to happen then is they're going to have to go somewhere, and they end up going through ATP synthase. The hydrogen ions, you can follow one of those in your book, the little red arrow, flows through ATP synthase, and that potential energy is used to drive the conversion of ADP to ATP. As that hydrogen comes through, it combines with oxygen, the same oxygen, and the reason why we breathe in oxygen is one of the reasons to drive this process. So the oxygen that's available from us breathing it in goes with the hydrogen and 2 electrons to make water. So at the end of the electron transport chain, we have formed water and we have formed ATP. ATP, that reaction happened because of that gradient. ADP reacts with the phosphate to make ATP. Now, this is huge. This is where most of our energy is made. So flip the page 136, figure 8.7. And so you need to know this as a summary of glycolysis and the Krebs cycle and the electron transport chain. They say in your book, here we follow the energy capture from one glucose molecule, which generates 2 molecules of pyruvate during glycolysis, that then enters cellular respiration. Cellular respiration is the Krebs cycle and the electron transport chain, which encompasses all of the reactions shown inside the mitochondrion. Notice that most of the ATP derived from glucose comes as a result of high-energy electrons donated by NADH and FADH2 to the chain, which also allows chemiosmosis to occur. Now, chemiosmosis is the formation of ATP. It's basically the last step of ATP production. So chemiosmosis occurs in the chain as the last step of ATP production. So what you're seeing on this figure 8.7, you see glucose. You know glucose is split during the process of glycolysis. The 2 main energy products of glycolysis are the 2 ATPs we made, as well as the 2 NADHs. Now, pyruvate enters into the mitochondrion and enters into the intermediate step. During that intermediate step, we made 2 NADHs. During the Krebs cycle, we made 6 NADHs and 2 FADH2s. The Krebs cycle also on its own produced 2 ATPs. Now, the NADH, the FADH2, goes into the electron transport chain. So you have NADH, so count them up. You have 2 NADH in glycolysis, 2 in the intermediate step, 6 in the Krebs cycle. So that's 2 plus 2 is 4, so 6 is 10. For every NADH made, 3 ATPs can be produced. So you have 10 NADH, you say that times 3, you get 30 ATPs made from NADH during the electron transport chain. For every FADH2 produced, you can have 2 ATPs made. So you have 2 times 2 is 4, so we made 30 so far, plus the 4 is 34, plus the 4 you see on the right-hand side, the 2 in glycolysis, the 2 in the Krebs cycle, ATPs that is. So we got 34 plus 4 is 38. So you see at the bottom 36 or 38 ATPs. There's a difference 36 to 38 just because sometimes the electron transport chain isn't as productive as otherwise it would be. So some books say 36, some say 38. But just to be on the safe side, they usually give you either or numbers. Now that is the end of cellular respiration. You're going to have to fill out the picture that you see on page 136 on your test, and you're going to have to fill out page 133. Page 133 and page 136 are crucial, And then on page 134, you have to be able to write that out in words, what's going on in the electron transport chain. Now, I don't have a slide for any information filled out on fermentation here, but I'm going to talk you through it. So they have in your text on page 136 what happens during fermentation. So just reading, virtually every organism on Earth uses glycolysis, and that's just a fact. About everything on Earth uses glycolysis to make energy. So why is fermentation necessary? Your next big heading, following glycolysis under anaerobic conditions. Now that's key. If oxygen is present, fermentation never even happens. But if oxygen is not present, right after glycolysis, you can't do the Krebs cycle. You can't do the electron transport chain. So right after glycolysis, if no oxygen is available, that's when fermentation occurs. Because you don't have the electron transport chain happening, you don't have nearly as much energy as made because you really just have glycolysis happening. Fermentation is required to convert NADH produced during glycolysis back to NAD, which is constantly recycled. So fermentation is a big thing to remember. It's under anaerobic conditions. Now where you see on page 137, some cells ferment pyruvate to form lactate. The fermentation of pyruvate to lactate is called lactic acid fermentation. Okay? Lactic acid fermentation happens in muscles that are working hard enough to use up all their oxygen. If you've ever gotten a Charlie horse before in your leg, where just a big pain kind of strikes you pretty hard, the scientific thinking is that lactic acid buildup in your muscles is causing that to happen. So your muscles will carry out lactic acid fermentation when you have no oxygen available. That's a big thing to remember there. Now flip your page, page 138, and you'll see some cells ferment pyruvate to form alcohol and carbon dioxide. And this doesn't happen in our cells. This happens in microorganisms, mainly yeast. So the yeast can carry out alcoholic fermentation under anaerobic conditions. Now with this yeast, you can just purchase yeast from the grocery store and add some sugar to it. And you can do a very basic alcoholic fermentation experiment. And you can smell, obviously, the alcohol from the yeast, the sugar, and you would need to add that to grape juice or some sort of juice substance. And if you let it sit over time, you can actually smell the alcohol coming off. The yeast that you buy at the grocery store is called Saccharomyces cerevisiae. And that Saccharomyces cerevisiae actually ferments the sugars in the juice and the sugar that you add in, and naturally produces alcohol, ethanol, and carbon dioxide. The bubbles that you see in any kind of alcoholic beverage are carbon dioxide from the fermentation, from the alcoholic fermentation by those yeast. There are other organisms that can carry out alcoholic fermentation and lactic acid fermentation. But this is really all you need to know about fermentation. You need to know that it's anaerobic condition. You need to know that lactic acid fermentation occurs in your body cells when you don't have any oxygen. And then you need to know the yeast cells can carry out alcoholic fermentation. And that's all you need to know for your test.